A nucleotide is the building block of a nucleic acid. It consists of a 5-carbon sugar covalently bonded to one of 5 different nitrogenous bases and one or more phosphate groups. The deoxyribose sugar in DNA has a naming system for its carbons. The nitrogenous base is covalently bonded to the first carbon. There is a hydroxyl group at the 3rd carbon, and the phosphate group is bonded to the 5th carbon. The nucleotides are bonded to each other with phosphodiester bonds between the phosphate group and the 3rd carbon of another nucleotide. The 5 different nitrogenous bases are adenine, thymine, cytosine, guanine, and uracil. Thymine, cytosine, and uracil are pyrimidines. Pyrimidines are characterized by one 6-membered ring. Adenine and guanine are purines. Purines are categorized by 2 rings, a 6-membered ring fused to a 5-membered ring. The nitrogenous bases found in DNA are adenine, thymine, cytosine, and guanine. The nitrogenous bases found in RNA are adenine, uracil, and cytosine, and guanine.
A strand of DNA has a 5' end and a 3' end. The 5' end is where the phosphate group is located, and the 3' end is where the hydroxyl group is located. DNA is synthesized from the 5 to 3 direction. The structure of DNA is meant to be anti-parallel, which means that the 2 complementary strands of DNA run in opposite 5 to 3 directions. So 5 to 3 and 3 to 5. Nucleotide bases have pairs. Adenine pairs with thymine. On RNA, adenine pairs with uracil. Cytosine pairs with guanine, and the bonds between the nitrogenous bases are hydrogen bonds. According to Chargaff's rule, the ratio of complementary bases, or A plus T and C plus G, are proportional to each other, which means that the amount of adenine will equal the amount of thymine, and the amount of cytosine will equal guanine.
Okay, moving on to DNA replication. Helicase is the enzyme that untwists the double helix of DNA at the replication fork into 2 parental strands that are used as template strands for replication. Here's the helicase. The replication fork is located on the DNA molecule at the end of the replication bubble, where the parental strands are being unwound by helicase. Single-stranded binding proteins, as you can see right here, bind to the unpaired DNA strands, preventing them from repairing, so they're pulling them apart. DNA replication can be categorized into the leading strand, as you can see right here, and the lagging strand.
The leading strand is synthesized towards the replication fork. Primase is an enzyme which synthesizes RNA primers continuously in the 5 to 3 direction, using the parental DNA as a template. RNA primer is created when primase adds RNA nucleotides one at a time to the 3' end. Here you can see that there are a total of 2 bases added. Once the RNA primer is made, DNA polymerase III starts to synthesize the leading strand continuously in the 5 to 3 direction as the fork progresses. DNA polymerase III is an enzyme which catalyzes the synthesis of new DNA by adding DNA nucleotides. It can only add nucleotides starting from 3' end. Here you can see that there are a total of 3 nucleotides that are added. As we mentioned earlier, the nucleotides have phosphodiester bonds between the phosphate group and the sugar. DNA polymerase I replaces all RNA nucleotides with DNA nucleotides.
Unlike the leading strand, the lagging strand is synthesized away from the replication fork. Primase does the same thing on the lagging strand, except it synthesizes RNA primers discontinuously as a series of segments. Primase adds 1 RNA nucleotide at a time to the 3' end, resulting in RNA primers. After the RNA primer is made, DNA polymerase III comes in and adds DNA nucleotides to the lagging strand starting from the 3' end. This process is repeated across a lagging strand, creating multiple fragments called Okazaki fragments, which are produced discontinuously. Here we have our Okazaki fragments, so we have 2 fragments. Here you can see the 2 Okazaki fragments in which one of the fragments runs into the primer of the next fragment. Then DNA polymerase I replaces all the RNA nucleotides of the RNA primers with DNA nucleotides. Another enzyme called DNA ligase joins the sugar phosphate backbones of all the Okazaki fragments into one continuous strand of DNA. So the ligase enzyme is going to come in right here and join these 2 Okazaki fragments together to create one continuous strand of DNA.